All right, hi everyone. This is our last lecture on the cell cycle. And so I sort of was alluding to it a little bit when I was introducing that mTOR paper, because we talked a little bit about tumor suppressor, or there's a tumor suppressor gene that paper mentions, and basically tumor suppressor genes are genes that normally slow down the cell cycle, right? And slow down cell proliferation. So we're going to talk about the cell cycle. We hopefully remember what the cell cycle is. I'll remind you, but then we'll talk also about a lot about regulators of the cell cycle. Okay. All of which, again, we're going to always sort of be drawing our attention to its sort of relationship to cancer cells, essentially. Okay. All right, so just a little bit of introduction, right? So it says self-reproduction is perhaps the most fundamental characteristics of cells, right? So we're talking about or thinking about cell division, we think about mitosis, right? If I say cell proliferation, basically that means cells that are undergoing cell division, right? All right, so we hopefully remember mitosis. All cells reproduce by dividing into two. Each parental cell gives rise to two daughter cells, right? And that basically completes that cycle of cell division. Obviously, this has to be carefully coordinated and regulated. If it's not, like I said, it leads to things like cancer. If that cell cycle was not regulated in terms of on the other end being that there's not enough proliferation going on, right? It could affect things like wound healing, growth, right? And things like that. Okay. So there are specific proteins that basically control the progression through the cell cycle, okay? So it says protein kinases. And again, we've mentioned the word kinases, especially in that cell signaling chapter, right? That kinases are proteins that phosphorylate other proteins. And when they do that, they regulate the activity of those proteins, okay? I already said this. Again, defects in the cell cycle are common cause of abnormal proliferation that you see in cancer cells. All right. Okay. So it says the division cycle or the sort of cell cycle in most cells basically consists of four coordinated processes, right? So cell growth, then there's DNA replication in there, right? Because before a cell divides, it has to replicate its DNA. Distribution of the duplicated chromosomes to daughter cells, that's mitosis, and then the cells actually divide. Okay, so that should all be familiar to you. In terms of, and again, we're focusing on eukaryotes here. It says in bacteria, cell growth and DNA replication take place throughout most of the cell cycle. And basically those duplicated chromosomes get distributed to those daughter cells in association with the plasma membrane. Obviously much simpler process than what we're going to talk about here. Okay, so that's all I'm going to say about bacterial cells in this lecture. Okay, so we are focusing on eukaryotes just like we did obviously in that last lecture as well. 4 phases of the cell cycle. So this is the review part. This is the part we should already know. And it's okay, maybe you don't remember exactly, but this should trigger some sort of memory. Okay, so 4 phases, M, right? Mitotic phase or mitosis, G1, S, and G2. Okay, so in the M phase, like I said, it's mitosis, nuclear division, and then it ends with cell division, which is cytokinesis. We remember that cytokinesis is not really part of mitosis, right? Interphase basically is the part of the time when the cell is not undergoing mitosis, right? And interphase is composed of G1, S, and G2. Okay, so again, this should be review. So in that G1 phase, gap 1, sometimes people say growth 1 in terms of what that G stands for. But basically, this is the interval between mitosis and DNA replication, okay? The cell is metabolically active, it's growing, and then towards the end of G1, it's going to prepare for cell division, right? So in S phase, synthesis, that's where DNA replication is going to take place. G2, gap 2, more cell growth, and now the cell is going to prepare for mitosis, which is going to happen after G2, okay? Now, you should be able to draw the cell cycle like this. We should totally understand and know all of the phases. But like I said, we're going to go further and really talk a lot about regulation of the cell cycle. Okay. And so obviously that's what most of the lecture is going to be spent on here. All right, so if we, I don't know, we'll start from G1 here, right? So here's our cell, just, this cell just underwent mitosis, right? Cell division and produced 2 daughter cells, okay? This is actually showing you here 1 daughter cell that's exiting the cell cycle. And so you can think about, and this is kind of, and I don't know, I'd probably come back to this at some point, but the way kind of this works within the body is that there's usually a pool of proliferating cells that sort of stay in the cell cycle, right, and produce more cells. When cell division happens, right, one of those cells usually stays kind of within that proliferative pool, right, or that sort of stem cell like kind of pool or stem cell pool. And then one of them may exit out here, and this one, think about as going on to develop and mature into a specific cell type, okay? So those cells that are dividing are stem cell-like, right? Whether they're, neuronal stem cells or skin stem cells, they might be slightly differentiated, but they have these sort of stem-like features, okay? Once a cell exits the cell cycle, then that's when it will differentiate further and develop into a very specialized cell. So when we think about neurons, right, those are super specialized cells. Neurons aren't able to proliferate. However, there are, 2 areas of the brain where you have pools of these sort of stem cells that can divide to produce new cells. Okay, so just kind of a little side note, and I don't remember if I sort of come back to that, but I just sort of wanted to mention that just because it's sort of showing this here. Okay, so when we say a cell is in the cell cycle, that means it's a cell that is dividing, right? Or it's a pool of cells that are actively dividing. Okay, they're going through these phases. If a cell doesn't have an intention to divide, it's not really in the cell cycle, and it's sort of showing you that here. Okay. All right, so G1, GAP1, growth 1, right? Metabolically active, it's doing its thing. It's going to get ready then towards the end of G1 for division, right? So then we have cell division, DNA replication, right? In the S phase, that's happening here. So you can see that DNA is replicated towards the end of S phase, right? G2, you're going to have some more growth going on. The cell then is going to get ready for mitosis or for cell division, which is going to happen here. Okay, so you should know all of these steps in order, know what's happening at each one. Okay, and now we're going to layer on top of this. All right. Well, let's talk just a little bit about timing, right? So it says the duration of the phases will vary considerably depending on what type of cell you're talking about. So budding yeast, right, can progress through the cell cycle in 90 minutes, okay? Early embryos have cell cycles of 30 minutes, and there's actually no G1 or G2 phase. Why? In early embryos, what's there a lot of going on? Just proliferation, right? So the cells just need to rapidly divide, and so those G1 and G2 phases are basically gone or shortened a lot, right? And so that's what this is showing you, that there's basically only M phase and S phase, because basically all these cells are doing is dividing, okay? And because that needs to happen rapidly early on in development, you need to have enough cells there to begin with for then that differentiation and the formation of tissues and organs can happen after that. But if you don't initially have enough cells, right, those other processes won't be able to happen. Okay, so that's an important point to remember, that in embryonic cells or early on in embryonic development, the cell cycles of these embryonic cells look like this, right? Where there is no G1 or G1 or G2, sorry. Okay, and, sort of in contrast to this, it says some cells in adult animals cease division altogether. And I sort of gave you this example already. It says nerve cells, right? Neurons, again, once they're in their fully differentiated, mature state, are not able to proliferate, not able to go through mitosis and produce more, okay? However, remember, there is a pool. There's 2 regions of the brain where there are a pool of neuronal stem cells, okay? And there is cell division that occurs, okay? So I think I do mention this towards the end of the lecture. Anyway, just in case I don't. Okay, so it says others may divide only occasionally, okay, that have to replace cells that have been lost. So again, it depends on what tissue type you're talking about, what cell type you're talking about in terms of how much cell proliferation is going on, right, and how quickly they move through that cell cycle. There's lots and lots of variation. Okay, So it says here cell cycle analysis, right? So in terms of studying the cell cycle, right, you need to be able to identify those phases. And so you can identify the phases of interphase by DNA content is one way that you can do that. So animal cells in G1 are diploid, right? They have 2 copies of each chromosome. Their DNA content is 2N. During S phase, that replication increases that DNA content to 4N, right? Because now it's been replicated and then that cell's going to divide. Remember, 2N is diploid, is your normal DNA content in a normal cell, not in a sex cell, right? We're talking about somatic cells. That DNA content, right, can actually be determined by basically incubating cells with a dye that binds DNA, right? So it's basically that fluorescent dye will just non-specifically just bind to the DNA, and then you can measure the amount of fluorescence, right? So that's what this is saying. So now you measure the amount of fluorescence intensity, and that's going to tell you, or the amount of intensity is going to correspond to the amount of DNA, okay? And there's a special machine called a flow cytometer that can basically sort these cells based on their fluorescence intensity to basically determine sort of how many or what percentage of the cells have this much DNA, what has this much DNA, okay? And so this is basically showing you here, right? It's showing the amount of DNA per cells and then how many cells actually have that. So you can make a sort of estimate in terms of, or kind of, like I said, this isn't, I guess you do get an actual number. So whatever, so we'll call it an estimate, right? Of how many cells are in G1, how many in S, and how many in G2 and M. Okay, so this is just a way of analyzing what cells. And then, like I said, the flow cytometer sort of sorts those cells. So then you could take those cells in G1 and study those cells in G1. You can then take those cells in G2M and study those cells. Okay, so it's sort of determining what cells are in it, but it's also sorting them for you. Okay, now, like I said, a lot of this chapter is basically talking about regulation of the cell cycle, okay? And progression of cells through that cell cycle is regulated by both signals on the outside of the cell, right? Extracellular signals, but also signals internally within the cell, okay? Now, remember, and maybe you remember this from when we talked about this even in Bio 111 or General Bio, that there are checkpoints, okay? So we'll kind of start off by talking about that. Okay, so here it says a major control point called start, okay, will control progression from G1 to S. Okay, this start control point was first described in yeast cells, and I will tell you that a lot of what we know about the cell cycle was done in yeast cells. They're very easy to generate mutants in these cell cycle regulators to determine what the function is. So there's a lot of information that we gained from studying yeast cells, okay? So once cells will pass this start, this major control point, they're committed to entering S phase and basically going through the whole cell cycle from there, right? So the idea is that a cell does not want to progress into replicating its DNA, which requires lots and lots of resources, right? If it's not going to go through and divide. So that's sort of the one main checkpoint. And usually once cells once cells get past this checkpoint, they will progress through. But there are other checkpoints as well. Okay? So the passage through that start checkpoint, though, is actually really highly regulated by external signals, right? Like nutrient availability, the size of the cell, okay? If there's a short, and again, think about yeast cells, these are single-celled organisms, right? So, a lot of their activities dependent on nutrient availability outside the cell. So if there's a shortage of nutrients, yeast will arrest that cell cycle at start, and they'll enter a resting phase. And then once nutrients become available again, they may re-enter into that cell cycle. Okay, so this is basically showing you this sort of start checkpoint here in yeast. Okay, so Again, you can kind of see that checkpoint's towards the end of this G1. And again, it's going to take into account, is there enough nutrients available? What about mating factors? What about cell size? Sort of checking in on all of that. If all of these are favorable, it will pass into S and basically progress through. If not, it's going to halt and enter a resting phase. Okay? Now, it says, in order to maintain a constant size, yeast must reach a minimum size to pass start. So that's what this is saying. It's checking cell size. We already said that pretty much. It says the small daughter cells of budding yeast will spend a longer time in G1 and grow more than the large mother cell, right? So a smaller cell that, again, one of these daughter cells is going to spend a longer time here and sort of be halted here to make sure that it's large enough to progress through, okay? And so this is basically showing you these budding yeast, right? And so you can see the buds coming off. These are the daughter cells that are produced. All right, now that was in yeast. And like I said, a lot of the details and how the cell cycle is regulated, we basically have used yeast as a model organism, okay? In mammalian cells, there is a restriction point or a checkpoint that is similar to start. Okay, but it's not called start. So again, I'll say it again. If I ask you about the start checkpoint, or we talk about a checkpoint in yeast, it's called start. Okay, in mammalian cells, there is a restriction point at the sort of the same point in time at late G1 that functions like start, but we're not going to call it start. Okay, and this passage through this checkpoint is regulated by growth factors. Okay, so if there are not enough growth factors sitting outside the cell, then the cell is not going to progress through this first restriction point in G1. So we can call it a G1 checkpoint, restriction point, whatever you want to call it. Okay, here we're calling it restriction point, but again, it's at G1. All right, but it functions the same way. Okay. So same thing, if appropriate growth factors are not present in G1, progression will stop and the cell will enter a resting stage. So what's happening in yeast and what's happening in mammalian cells is the same. It's just that we're not going to call this checkpoint start in mammalian cells. And what's the sort of deciding factor outside of the cell in mammalian cells is growth factors, not nutrient availability, not cell size. It's really growth factors that are going to control this, okay? In yeast, we talked about cell size and nutrient availability as being those external signals. In mammalian cells, it's growth factors. Okay, so hopefully we can kind of distinguish between the two. And if you have your, you should have your review sheet in front of you, maybe that's something you might want to write down, a little comparison between start and restriction point. So start in yeast and restriction point in mammalian cells, some similarities and some differences. Okay, all right. So it says here, skin fibroblasts are arrested in G0 until they're stimulated by PDGF. I think I mentioned PDGF maybe in that cell signaling lecture, platelet-derived growth factor. So these skin fibroblasts are only going to be stimulated to proliferate if there is a cut, right, or a wound, okay, to repair. So they're sort of sitting there waiting for this growth factor signal to then basically have them enter into that cell cycle and progress through, okay? All right, so this is what this is showing you, restriction point. Again, growth factors is what's going to regulate or sort of be the indicator as to whether a cell should go past this restriction point and enter S phase and then basically progress through and divide, or if it should enter G sub 0, that resting state. And remember again, they then can enter back into the cell cycle. Just like that example we gave of the skin fibroblast that might be hanging out here in G0, once PDGF is available or they sense that in the extracellular environment, they will enter back in and progress through, okay? All right, now, there are, like I said, other checkpoints as well and other sort of control mechanisms. So it says some cells are controlled in G2, okay? A specific type of fission yeast is actually controlled by transition from G2 to M, right? So that's the point where cell size and nutrient availability will be monitored, not at that start signal like we talked about before, okay? So that's what this is showing you here. Okay. G2 to M. All right, so anyway, there would be a checkpoint here where it's basically checking to see if there was enough nutrients available. Okay, so this is basically showing you the sort of cell cycle and the division of fission yeast. To be honest, I'm not, that's not really something that I'm that concerned with too much. But the point is, the reason why we're bringing this up is to sort of kind of, tell you that we sort of figured out or discovered this sort of other checkpoint in this type of yeast, right? And then now we're going to talk about eukaryotic cells, not eukaryotic cells, mammalian cells again, or other vertebrate cells where you actually see the same type of control. Okay, so but again, I sort of want you to really understand the start and the restriction point that we talked about, and now understanding sort of that you can control the cell cycle at other points as well, not just at that start or that restriction point. Okay, so that's sort of the take-home message. I gave you an example, just mentioned an example in another type of yeast where you see that type of control. Okay, but there are also other cell types where you see this as well. So it says in animal oocytes, right? So you'll see cell cycle control in G2 as well. So for instance, vertebrate oocytes, right, will remain arrested in G2 for long, long periods of time, right? So if you think about, egg cells in humans, right, they remain arrested in that G2 phase, okay, until there is some sort of external signal that triggers them to basically progress through, right? So that's the example that we're giving there, right? So progression to M phase is triggered by hormonal stimulation, okay? All right. Now, the other thing to kind of think about is coordinating the steps of the cell cycle, right? Or the phases of the cell cycle. The cell has to have extensive mechanisms to make sure that these phases are happening in order, right? The cell can't go through G2 before it went through G1 or before it went through S phase. Obviously, these things only function in order, okay? Right? So obviously the cell, another example, right? The cell can't go through mitosis before DNA replication has occurred, okay? So I think you understand why these things have to be done in their appropriate order. And so that's why I tell you, you need to know the order, right? So if I ask you about the cell cycle and tell you to, describe the phases, they better be in order, okay? All right. So coordination of cell cycle phases is dependent on a series of cell cycle checkpoints, right? So what we were talking about before was sort of these, right? We mentioned sort of control the cell cycle, meaning kind of are cells in the cell cycle or they're not, right? We talked about start and restriction point, which are both at the end towards the end of G1. And those are really, like I said, is the cell going to go into resting phase or is it going to stay in the cell cycle and progress through? We also then gave you an example of cells that sort of do that at G2 instead of during G1, okay? But now the next part that we're talking about are these checkpoints. And again, when we went over this in General Bio or in Bio 111, we sort of showed the cell cycle and said, here are the checkpoints, okay? So I think we've sort of already talked about that first one. Now we're going to talk about what these checkpoints are, is kind of through the cell cycle to make sure the cell is going through these phases in order, and also to make sure that what was supposed to happen at that phase has happened, okay, before it moves on to that next phase. All right. So just to kind of distinguish as to why we're sort of calling these something different and sort of talking about them a little bit separately, okay? So again, this is all about coordinating those phases, making sure they're happening in order, making sure everything that was supposed to happen during that phase did happen before it moves on to the next one, okay? So that's what this is saying. They prevent entry into the next phase until events of the preceding phase have been completed. If those events have not been completed, it is important that the cell does not move into that next phase, okay? So these are cell cycle checkpoints here. So there are DNA damage checkpoints and there's a spindle assembly checkpoint. Okay, so these are the checkpoints that you've sort of mentioned before, or I've mentioned them before, like I said, very briefly in Bio 111. Okay, so DNA damage checkpoints do exactly as they say. They ensure that damaged DNA is not replicated and passed on to daughter cells. Okay, so that cell cycle would be arrested until that DNA is repaired. If it's not able to be repaired, then the cell will be sent into apoptosis, right? And will go through cell death, okay? Because again, we don't want damaged cells to keep dividing, right? Spindle assembly stops mitosis at metaphase if the chromosomes are not properly aligned. Again, if those chromosomes are not properly aligned, those daughter cells that end up being formed are going to have genetic defects. So we want to try to prevent that, okay? And so that's these here. So again, the start and the restriction point that we talked about here, think of that as being kind of something different that's going to check for external signals like growth factors, like nutrient availability. We also then mentioned another type of checkpoint like that in a different type of yeast in where it happens in G2. Again, checking for nutrient availability, okay? Now these checkpoints here are all there to make sure that the cell is progressing through the cell cycle properly, okay? So if you notice here, this checkpoint is checking for DNA damage, okay? Again, in a similar spot as that restriction point that we talked about. But again, think about restriction point is checking for the presence of growth factors. This is checking for the DNA damage, okay? So these are DNA damage checkpoints, 3 DNA damage checkpoints. And then there's this spindle assembly checkpoint, okay? So here again, it's not gonna move in, it doesn't make sense for it to move into S phase and replicate damaged DNA, okay? Again here, after S phase occurs, it's possible that there might be, mistakes that were made by the DNA polymerase, and it's important that they get they get fixed, right, before the cell would go through division, okay? So it's important to check for DNA damage after replication, and there's sort of then one more check here to make sure anything that should have been repaired, maybe if it was halted here, was just one last check to make sure there is no DNA damage before we send the cell into division, okay? Again, spindle assembly like I talked about, okay? So these are the checkpoints. So if I ask you for the checkpoints, or I say 4 checkpoints, then it's these 3 DNA damage in the spindle assembly. If I'm going to ask you about, start or restriction point, I would refer to that as, or how does the cell, a mammalian cell, check for the presence of growth factors or something like that? What is the sort of regulation point, which would be restriction point, right? And again, that is whether the cell really is going to stay in the cell cycle or not. Again, these are to ensure proper progression through, check to make sure everything that was supposed to happen during this phase has before it moves on to the next. Okay, so you can kind of see how there's different function between these checkpoints and what we talked about before, that start or that restriction point, okay? All right, now we got to talk about regulators, okay? Regulators of cell cycle progression. So great, awesome, we got checkpoints, right? Making sure things are happening so that it moves through, but how the heck does the cell move through, right? How, what makes this phase different than this phase? What's around to make this different? Also, how is this regulated, right? I mentioned tumor suppressor genes, right? And we said if a tumor suppressor gene is mutated and the cell cycle is increased, right? Or the activity of the cell cycle is increased, it leads to cancer, right? So how does the body or the cell regulate this? How does it keep it in check to make sure that cells are proliferating to the level that they should? Because again, overactive proliferation will lead to cancer. Okay, so you might imagine that there are lots of regulatory mechanisms to control this, to which are super, super important. Okay, so that's what this next part of the lecture is about. And you can see at the top, regulators of cell progression, right? Or regulators of the cell cycle. It says here, a conserved set of protein kinases. Okay, so these protein kinases will trigger the transitions into those different phases that we talked about, okay? Now, you're gonna have to bear with me here, but it's important to go through these experimental approaches that were used to identify the cell cycle. So we're gonna follow along here. Each one is gonna provide a certain piece of information that is super, super important, and that was super important in our understanding of regulation of the cell cycle. Okay, so so I guess get ready. If you need to pause it for a second, go get a drink of water. You can do that. Actually, I need to do that for 1 second. All right, so let's continue on here. So again, this is going to describe the experimental approaches that basically allowed us to identify cell cycle regulators. Okay, so number one, studies of frog oocytes. Okay, so we mentioned this before, right? Of vertebrate oocytes that are arrested in G2 until there's some hormonal trigger to tell them to go into M phase. Okay, so frog oocytes, again, arrested in G2 until hormonal stimulation triggers them to enter into M phase. Okay, and so basically, and these are older studies, right? 1971, way before you guys were born. Researchers found that these oocytes could be induced to enter M phase by basically injecting the cytoplasm of these cells, okay, of these oocytes, from oocytes that had been hormonally stimulated, okay? So they have oocytes that are not hormonally stimulated. They have another set of oocytes that have been hormonally stimulated. So they've already been the hormone that triggers those cells into M phase, they've already been exposed to, right? And so if they take they take some cytoplasm from those hormonally injected oocytes, and they inject that cytoplasm into those oocytes that have not been exposed to the hormone, okay, they basically can see that those cells could be induced to enter M phase just by having the cytoplasm from those other cells. Okay, so I'm going to show you a picture. And so they called this cytoplasmic factor maturation promoting factor, or MPF. Okay, so again, here's this first set of experiments. And again, it wasn't just one group that did it, okay? But this is sort of, again, a summary of these early experiments in frog oocytes, okay? So again, there are oocytes that basically were exposed to progesterone or injected with progesterone, that hormone that's going to induce them to go into M. So you see this, right? G2 exposed to progesterone, they go into M phase. That's the hormone that's going to basically stimulate them to go into M phase. If you take cytoplasm from those induced cells and you actually just inject or micro-inject, right? You're using a very careful micro-injections using a glass pipette to inject into a cell that's just in G2, no progesterone around here, you can see that then causes them to enter M phase. So think for a second what that tells you, right? What does this experiment tell us? Right? It tells us that there is some factor present in this cytoplasm. So again, the progesterone, let's call it this way, the progesterone never entered the cytoplasm. I guess that's not really true, but whatever. Let's just think of it like that. The progesterone just stimulated these to go into M phase, okay? And so the idea is that there is some factor present in the cytoplasm that's able to induce these cells, okay? It's not progesterone, put it that way. We know it's not the actual hormone. It's some other factor present in the cytoplasm. So it's this secreted factor that's there in the cytoplasm, okay? And so scientists called this MPF. They don't know what it is at this point, right? They just know there's something there that can basically induce cells to move from G2 to M, okay? So they identified this MPF. They called it MPF. Now, other work showed that MPF is also present in somatic cells. Okay, so it's not only in these oocytes, you also see MPF in somatic cells, where it also induces entry into M phase. So suggesting there's this general factor that exists that basically is responsible for moving cells from G2 to M. Okay, that's what this says here. MPF appeared to act as a general regulator of the transition from G2 to M. Again, think about why this is important. We're trying to understand the cell cycle, how cells move through the cell cycle, right? All right, so now we're moving on to number 2, right? We said there was 3 experimental approaches. So the first one was those Frege oocytes, basically determining that there was some factor in the cytoplasm that can induce cells to move into M phase. They called that factor MPF. All right, number 1. By the way, you guys should know all this. I'm just saying you better be writing this down on your review sheet. I will certainly ask you about this on the final exam. Number 2, genetic analysis of yeast. Okay, so now we're using yeast cells. That was Frege oocytes, the last one. Investigators found that temperature-sensitive mutants, okay, so temperature-sensitive mutants that were defective in cell cycle progression, okay, so these are mutants that weren't able to progress through the cell cycle, okay, because they don't have these CDC genes, okay. So they basically determined that these CDC genes are required for passage through start and entry into mitosis, okay? And then basically figured out that these CDC genes actually encode protein kinases, okay? So, but what this is showing you is basically if those CDC genes are mutated, okay, they're not able to progress through, okay? But when they then are able to sort of turn on those genes and they're functioning properly, they'll be able to progress through. Okay, so don't worry about this, but there is a, I guess, experimental approach where you basically can turn sort of on and off things using temperature. Okay, so this is the way that, so basically, again, this is mutant CDC genes, non-mutant. Okay, all right, that's how they're sort of turning it on and off. So what does this tell you? It tells you that you need to have functioning CDC genes enable for that cell to be able to progress through that cell cycle, suggesting again that to particularly move through start, right? So this indicates that these CDC genes are important for progression through start and basically progression through the cell cycle. Okay, so remember the start is that point where in yeast cells, it's checking for nutrient availability, right? And cells that get past start usually progress pretty much through that cell cycle, right? So what's responsible for that, what enables that to happen, okay? All right, so this is basically number 2, is identifying specific genes, okay, in yeast that are required, okay, for these cells to move through the cell cycle, okay? So then what do those genes code for, right? And are those genes also found in other eukaryotes? And the answer is yes. And so other studies showed that these similar genes, essentially related genes, were found in other eukaryotes as well, okay? And again, we said they code for kinases, right? So it says the protein kinase has since been shown to be a cell cycle regulator conserved in all eukaryotes known as CDK1, okay? So this is one of the main regulators of the cell cycle. So we can think about, we showed this experiment using yeast cells with start, but if you mutated these types of genes in a mammalian cell, right, they wouldn't be able to progress through that restriction point. Remember, we just used different terminology for there, okay? So conserved, these same types of genes. So CDK1, okay, was identified. All right, so we move on to number 3, next experimental approach, okay? So now we're getting, we're in 1983, we started in 1971, okay? Basically, this group now used sea urchin embryos, okay? And they identified 2 proteins, which are 2 types of cyclins, that will accumulate throughout interphase, right? But they rapidly get degraded at the end of each mitosis. So it suggested that there's some role of these proteins, these cyclins, in inducing mitosis because they're there, but then they rapidly get degraded essentially before a cell moves through mitosis, okay? And so it says here, later studies, so now we're going back to our first experimental approach, showed that microinjection of cyclin A, right, so one of these cyclins that they found, into those phagocytes basically will trigger them to move from G2 to M. So now, if we go back here, instead of taking cytoplasm, right, here they're going to inject these cells with that cyclin A that was discovered, and basically this will induce them to do the same thing, okay? All right. And so this is basically showing you what happens in terms of, and there's other cyclins as well, okay? And so there's sort of a cyclin for each phase, kind of. So cyclins, this is just showing you this cyclin A here, right? Kind of showing you between interphase and mitosis, that basically you see it's rising up, and then basically as you get through mitosis is completely degraded and then rise. So that's why they're called cyclins. And so you basically see the levels increase and then dramatically decrease as they're degraded, okay? So this is basically from kind of these sea urchin studies, some of the things that they noticed, okay? So they noticed this, and then they were able to figure out what that was, okay? So they identified two particular cyclins, okay? One of those, cyclin A, like I said, if you inject them into those phagocytes that we talked about in the beginning, right, you actually can see that those would induce them to move from G2 to M, okay? All right. So now we're in 1988, right? 3 experimental approaches converged, basically, when MPF was purified. Okay, so now we go back to here, right? There's this factor that existed in the cytoplasm that can induce these cells to move into M phase. In 1971, we didn't know what it was, right? We just know it was some factor that did that. Well, now when you purified that MPF and studied what it's made up of, it's actually composed of CDK1 and cyclin B. Okay, so again, a cyclin. So we were kind of talking about, we mentioned cyclin A here, but regardless, the main point is actually this CDK, okay, and a cyclin, okay? Like I said, there's many different types of cyclins, and they work actually hand in hand with these CDKs, okay? So there's other types of CDKs as one, but bottom line is that MPF is basically composed of these two things that were discovered later, right? CDK and a cyclin, okay? So that MPF, which we didn't know about in 1971 what it was, we just knew it could induce progression from G2 to M. Now, in 1988, basically, if you study that MPF, because now we have technology to do that, right, at this point, to determine what it's made up of, we know it's made up of CDK1 and cyclin B. Also, we are already CDK1 and cyclins had been discovered, right? And so now we know that the MPF is actually made up of that, okay? So cyclin B is a regulatory subunit required for catalytic activity of the CDK1. So I'm going to show you what kind of all this kind of coming together here, all right? All right, so there's your MPF. So if I ask you what is MPF made up of, you will tell me cyclin B and CDK1. And like I said, don't be confused with the fact that I mentioned cyclin A here, okay? Let's just kind of consider that as being, sort of general cyclins, okay? they like I said, they work hand in hand with these CDKs. So they need to be, their sort of regulation is really controlled by the CDK. All right, anyway, so we'll move on from there. Okay. So it says, further studies demonstrated that regulation of MPF by phosphorylation and dephosphorylation of CDK1, okay? So cyclin B is synthesized and forms complex with CDK1. I'm going to show you a picture, okay? CDK1 is phosphorylated and inhibited, leading to accumulation of inactive CDK1 cyclin B complexes during G2. That's all very confusing, isn't it? Dephosphorylation activates CDK1, which phosphorylates several proteins that initiate the events of M phase. So there's lots of phosphorylation and dephosphorylation going on. Like I said, the CDK1 is really the controller of this, okay? And so after this sort of series of phosphorylation and dephosphorylation that's regulating the activity of CDK1, cyclin B will be degraded, okay? And then once cyclin B is degraded, that CDK1 becomes inactive, and basically the cells will exit mitosis. Okay, and so this is what I was referring to. Now, nobody needs to memorize when it's phosphorylated and dephosphorylated or anything like that. This is kind of the same idea as I said with the cell signaling lecture, in terms of understanding what's going on here. Okay, we don't need to memorize any of these players in here. We just need to sort of understand this process. And I will tell you again, like I mentioned, there are different cyclins, there are different CDKs, but all of them sort of in terms of different phases, right? Right now we're kind of talking about moving from G2 to M, but if you think about moving from G1 to S or from S to G2, similar mechanisms are in place, but with just different cyclins, okay? Again, it's about sort of just understanding what's going on here, not memorizing any of this. So in that previous slide where in words it kind of says, well, this is phosphorylated, then it's dephosphorylated, right? This is what it's referring to here. So you can see, again, this is complex, right? There's a lot going on. But again, important to understand this because if we're going to talk about, targeting or therapeutic interventions for cancers, this might be a place where we might want to do it, right? And so we need to know all of those molecules involved in the pathways in order to target it, right? So here, for instance, this is basically showing you, this WEE1 is going to phosphorylate this CDK, right? And it's particularly showing you what amino acid is phosphorylated on. Then you can see the CDC25 will dephosphorylate, right? So there's other proteins even involved, even in this sort of simplified picture, besides the cyclin and the CDK, okay? And so it's phosphorylated, then it's dephosphorylated. At that point, this MPF, which is really this complex, is active, okay? And that will then that cell will go through mitosis, okay? But eventually, that cyclin B will degrade, and if this CDK1 is not bound to the cyclin, then none of this is going to happen. So until it then rejoins with another cyclin, that process will continue, okay? So at the end of mitosis, is totally degraded, and basically we're kind of moving through this here, okay? So you can see here, there's another, and it ends up getting dephosphorylated. And until it's dephosphorylated, it wouldn't be able to then form another complex with cyclin B. Okay, so phosphorylation is greatly regulating the activity of this CDK1. And so you can see I kind of said that CDK1 is the one that sort of controls the cyclin. Or it's kind of I guess what I meant is sort of it's the one kind of doing everything, right? It's the one that's getting phosphorylated. And so if it's not phosphorylated and dephosphorylated in the right way, then, it's not going to trigger that cell to move through mitosis. Okay. So this is when it's in this active state. So it needs to be complex. So cyclin B and CDK1 need to be in a complex. CDK1 needs to be phosphorylated appropriately in order for this to occur. Okay. And so you can see there's various points kind of in this pathway where things can be regulated. Okay. From here. All right, so it says, further research has established that CDK1 and cyclin B are members of protein families. And so, like I said, there are many cyclins and CDKs, okay, that will control different phases or different sort of transition points through different phases, right? So like I said, moving from G1 to S or from S to G2, okay? So that's what this is telling you here. Says here in yeast, right? CDK1 controls G2 to M transition in association with mitotic B type cyclins. Okay, so that cyclin B that we're talking about. CDK1 controls passage through start and entry into mitosis in association with G1 cyclins. Okay. All right, so this is illustrating the point where there's different cyclins involved, right? So here, B cyclins, G2 to M, G1 cyclins, if we're talking about moving through start, okay? So it's still CDK1, but it's going to associate with a different cyclin. In higher eukaryotes, there are multiple cyclins, like I said, and multiple CDK1 related protein kinases, okay? So again, there are multiple CDKs, there are multiple cyclins. Depending on what phase transition you're talking about, you might have a different cyclin and a different CDK, okay? But this sort of same basic mechanism or sort of kind of set up here is what's going to happen at those different transition points. Okay, so the idea is understanding that if I asked you sort of what proteins are involved in controlling the cell cycle, right? It's CDKs and cyclins. Okay, and I just went through with you how these were discovered. Okay, so it's important to understand that process. So it says complexes of cyclins and cyclin-dependent kinases, right? So showing you some different ones that I just talked about, right? So there are many different CDKs, there are many different cyclins. So if we look at yeast, a little more simple, right? You can see some of these different cyclins associating with CDK1 here, different cyclins associating with CDK1 here. So kind of in the yeast cell, it's showing you that it's kind of one main CDK that's involved that then associates with different cyclins depending on what phase transition you're talking about. In animal cells, you're going to see that there are different CDKs as well as different cyclins. Okay. So you can see that here. Okay. So you can kind of see how this, we said if you put cyclin A, if you injected cyclin A, that it caused transition here. You can see that here. As you move through, okay? It says studies of CDKs and cyclins basically revealed that CDK is available or capable of substituting for all the other CDKs, right? So if we genetically modified mice, right, and we basically got rid of other CDKs, but left the cyclins intact, CDKs can basically compensate for one another, right? So based on a lot of these studies, it was basically showing if you got rid of pretty much all the other CDKs, right? Some of these other CDK4, right? CDK2, but CDK1 was left intact and all the other cyclins, basically CDK1 can compensate for the activity of the others, all right? So I don't know if you want to think of CDK1 as sort of being the most important one. And again, you can kind of see that in yeast, that's the one that's involved, right? And then we sort of, in animal cells, are sort of evolved to have other CDKs available as well. All right, so moving on here. Because obviously it's not that simple, right? It never is. There's definitely more in terms of the regulation part of it. And so anyway, it says the association of CDKs and cyclins, right? So this is activity now. So activity of CDKs is regulated by 4 mechanisms. So we kind of just saw from that one figure I showed you, to which I told you nobody needs to memorize, right? That you can see that CDKs are regulated just by their association with their appropriate cyclin, okay? So they have to form these complexes. So that's one thing, right? So again, this is showing you CDK and its association with cyclin. There's also something else in there that we're going to talk about in a second, okay? It says activation of CDK cyclin complexes requires phosphorylation at threonine at position 160. If we go back, well, all right. I guess it's kind of showing you it here, but anyway, I must have another picture. Oh no, I don't. All right. Anyway, if you go back to that one figure that I showed you, I think I pulled out some of these figures because I didn't want it to get confusing and have too many different figures. But if you go back, you'll see that there are specific positions, and I kind of pointed this out, where the CDK must be phosphorylated, right? So the point is if it was phosphorylated on a different position, a different threonine, right, or another amino acid, that's not going to be okay. It has to specifically be phosphorylated on that one specific threonine, okay? So it's saying here, activation of CDK cyclin complexes requires phosphorylation of threonine at position 160, and that's actually catalyzed by a specific another specific kinase, right? So CDK activating kinase, which is composed of CDK7 and cyclin H. Okay, you can see how complex this is. So there are other proteins in that one figure that's specific for movement into G2, from G2 to M, sort of, it shows you that, right? That there's other proteins involved there that are going to be responsible for the phosphorylation part. And so those proteins are just as important as the CDK and the cyclin, really, okay? So this is kind of another activating kinase, which is composed of another CDK and another type of cyclin that will phosphorylate, all right? It says inhibitory phosphorylation of tyrosine near the CDK amino terminus is catalyzed by Wee1 protein kinase. So we mentioned there's Wee1 in that other figure, right? It says the CDKs that are then activated by dephosphorylation by CDC25. Okay, so if we go kind of back to that figure, you can see some of this going on here. Okay, so here this is showing you E1 phosphorylation. This is showing you dephosphorylation by CDC25. Okay, I mean, I guess we should know, and again, I would show you this figure if I asked you this question though, but if I asked you what the function of CDC25 is, we should be able to pull out from this figure that basically it's removing phosphates, right? It's pulling off these phosphates, it's dephosphorylating. Whereas WEE1 is adding phosphates, okay? It's phosphorylating, right? You can also say down here, this APCC complex is phosphorylating as well. And so that's that initial phosphorylation that we were talking about, okay? All right, so anyway, so again, nobody needs to memorize that. And again, some of the specifics in terms of what's phosphorylating what, right? In terms of like WEE1 or CDC25, some of those players will vary depending on what cyclins and what cyclins and what CDKs we're talking about, right? Depending on what transition point we're talking about, right? If so, again, we were focusing sort of on the G2 to M transition and looking at that specific mechanism. But it's going to be different, again, depending, right? Because remember, if you look at this picture here, right, we can see that there are many different types of CDKs and cyclin complexes, okay? And so the specifics of those mechanisms, right, if you looked at a diagram for you know, one of these compared to the one we looked at, things are going to be slightly different, okay? But in terms of main kind of things that will regulate the activity of CDKs, I want you guys to understand this. So the one main thing, association of CDKs with cyclins, super important, okay? CDKs don't work alone, cyclins don't work alone. They have to form that complex in order to function, okay? And then you can kind of see that example from that diagram that I gave that we looked at, okay? All right. Activation of cyclin CDK complexes requires phosphorylation, okay? If that initial phosphorylation doesn't occur, then it's not going to be active, okay? You also then need this inhibitory phosphorylation, okay, by WEE1. And then you're going to need to be, then it needs to be dephosphorylated before it's completely active. Okay, so there's a lot of regulation points that go on, and we're not going to get into all the whys. I don't think we know every single why in terms of why is it phosphorylated first, then dephosphorylated, why, you know what I mean? Why is all that happening? There's probably many reasons, okay? Another thing that we didn't talk about just yet is that there are inhibitory proteins called CKIs that can inhibit CDKs, right? So they're called CDK inhibitors, right? And so if a CKI binds to a CDK, it's going to inhibit it, okay? So there are different types of CDK inhibitors. Give you some examples here. Mammalian cells, there's an example of CYP, INK4. And so, you know, these are just some families of inhibitors and some examples here, okay? So in terms of what so the CYP family, right? Some P21, P27 are the sort of gene names for some of these separate ones that are a part of the CYP family, and what cyclin CDK complexes they will inhibit, okay? And if they're there and they bind to that complex, they're going to inhibit these phases, okay? So that's another level of regulation. So kind of think about like if these inhibitors come in, it sort of overrides all the other kind of things that we talked about here, right? So this is, again, what these four things we're talking about here is how our CDK, or how is CDK activity regulated, okay? So we gave four mechanisms. Association of CDKs with cyclins, first one. Phosphorylation, right? And then basically phosphorylation and dephosphorylation, so more. So it's kind of, I guess you can kind of think of, you could probably say association with the cycling complexes, right? Phosphorylation and dephosphorylation, you can sort of say in general, I guess, right? And then number 4 would be binding of inhibitory proteins, CDK inhibitors. And so if you see the term CDK inhibitor, you should know what it does. It's going to inhibit the CDK. If the CDK is inhibited and not active, the cell will not be able to progress through that phase, okay, and move into that next phase. And again, there are specific types of inhibitors that will affect specific types of CDK cycling complexes, okay, and then therefore affect different phases. All right, so I hope this sort of makes a little bit of sense. Again, complicated, right? You know, I only showed you that sort of one diagram to kind of keep things simplified, understand that there will be variations in what's happening there, but the sort of main mechanisms are similar, okay? All right. So it says the combined effects of these modes of CDK regulation are responsible for controlling cell cycle progression, right? In response to checkpoint controls in extracellular stimuli. So we talked about first the restriction point, right? Then we talked about cell cycle checkpoints. Those are checking to make sure things are going on properly. Then if they are, right, these cyclins and CDKs will sort of be initiated in terms of actually doing the work to move those cells through the cell cycle, okay? So the checkpoints are checking to make sure everything's okay, relaying that information to these cyclins and CDKs that actually, again, are the sort of doers to move cells through. If that sort of helps kind of put things into a bigger or broader picture, all right? Okay, says here, proliferation of animal cells is regulated by extracellular growth factors. We already said that, right? That control progression through the restriction point in late G1. We said that, okay? This implies that intracellular signaling pathways ultimately act to regulate components of the cell cycle machinery. Okay, yeah. So there are specific type of cyclins that are sort of a link between growth factor signaling and cell cycle progression. So we can kind of think back to last chapter, right? We have the idea that growth factors sit on the outside of the cell, right? They bind to their specific receptors, okay? And then how do you then link sort of then what's happening or what's initiated by the binding of the growth factors to the receptor to changes in the cell cycle? Okay, so that's kind of think about that. That's what this is saying here. So these D-type cyclins are involved in linking that, okay? So it says growth factors will stimulate cyclin D1 synthesis, okay? So this is a pathway that I think we mentioned in the last chapter, RAS, RAF, MEK, ERK, right? So once this growth factor bind stimulates this pathway, cyclin D1 will ultimately be stimulated, which will then affect the cell cycle, okay? So cyclin D1 is only synthesized if growth factors are present. So now we're talking about a specific cyclin, okay? A specific cyclin that's going to be involved in moving cells through the restriction point, okay? We talked about, back here, this example was talking about MPF specifically, okay? And basically, you know, thinking about start. Let's think about start here, okay? All right. Okay, so let me go back, sorry. All right, so now we're specifically talking about growth factors in animal cells, okay? Again, if we think back to last chapter where we had, whatchamacallit, we had, no, I guess this isn't start. Sorry, I just didn't realize I said something wrong. Think about this here, I'm sorry, as what's regulating progression from G2 to M, right? Sorry, it's not start, right? This is particularly specific to this initial, sorry to keep flipping, where am I? Right? This is what we started with. We're talking about progression to G2 to M. And again, why did we start here? Because this is easy to observe, right? And this is where we started with injecting the cytoplasm. So this specific mechanism that we were showing here, okay, in terms of this diagram, is specifically talking about MPF, right? Which again is that factor that was discovered first that could cause cells to move from G2 to M. And then now this is showing you specifics, right? We said what makes up MPF, and then how does this then progress cells through, right? How is it sort of regulated? Okay, so this is G2 to M. I don't want to confuse anyone by saying the wrong thing, duh. All right, now this next part we're going to talk about is cyclin D1 particularly, and actually coupling growth factor signaling, right, to cell cycle. So again, if there's a lot of growth factors around, cells are going to proliferate, right, be induced to proliferate. So this is basically showing you growth factor binds to its receptor, right, and then initiates this pathway that we talked about last chapter that then leads to the synthesis of cyclin D, right, D-type cyclins, that then will progress cells through the checkpoint. So if we're saying it's going to stimulate proliferation, right? We kind of had that. Remember, we had this sort of side-by-side comparison of growth factors and cytokines. Growth factors will stimulate proliferation, cell cycle. How do they do that? One of the big players and how they do that is by producing these D-type cyclins, okay? And so again, you can see that these cyclin D, these D-type cyclins are now associated with a different type of kinase. But in terms of what's happening and how that progression happens, go back to that. I won't flip back again and drive you crazy. But if you go back to that first diagram that I showed you some of the specifics, right? That same kind of thing is going to be going on. All right. So again, I could have put in more diagrams like that first one that I showed you. However, I felt like that would end up being confusing, okay? And so I left just that one in there where we're talking about G2 to M just to illustrate some of the specific mechanisms that are happening in terms of that phosphorylation, right? And dephosphorylation and how the CDK and the cyclins work together, okay? And then also showing that cyclin being degraded once that cell moved through mitosis. Those general mechanisms that are happening there are happening no matter what you're talking about. So even talking about this point here, moving through the restriction point and involving CDK4 and the cyclin D, similar mechanisms are going to happen in terms of the phosphorylation and the dephosphorylation, and that cyclin D will ultimately be degraded once that cell passes through. Okay, so that was meant to be illustrative and is representative in general of things that will happen with all of these CDKs and cyclins. Okay, but specifics will be slightly different. All right, hopefully that helps make a little bit more sense. All right, it says cyclin D1 is also rapidly degraded by that APCC. Again, you saw that in that first diagram. Okay, and so it says intracellular concentration will fall rapidly if growth factors are removed. Super important, right? It can't be hanging around long and keep stimulating proliferation when really there's not a lot of growth factors out there. If there's a growth factor there, it initiates this process and then it's rapidly degraded. If there are more growth factors there, then the same thing will happen again, okay? So that's what this is saying here. As long as growth factors are present, okay, that basically cyclin D1 will be made and will still be pushing cells through. But when growth factors become unavailable, this will stop, okay? Cells will not progress through that restriction point. It says, defects in cyclin D regulation can contribute to the loss of growth regulation, which is a characteristic of cancer cells. Because again, think about this, because cyclin D is particularly that cyclin that's responsible for the progression from G1 into S, which is the main restriction point, okay? And so if you lose that regulation, cells will just enter into that cell cycle and progress through and divide when they shouldn't, okay? So many human cancers arise as a result of defects in cell cycle regulation. I said that a bunch of times and I'll say it one more time, okay? So here is some more about that and some talk about tumor suppressor genes, okay? And so we've mentioned these before. So here's one example. So RB, okay, retinoblastoma, right, is a substrate protein of CDK4 cyclin D complexes. Okay, so again, CDK4 cyclin D, this complex is responsible for moving cells through that restriction point. Okay, and again, we said if most cells that move past that restriction point will actually go through the whole cell cycle and divide. Okay, so it's really important. Cyclin D will only be produced if there are growth factors available. Okay, and again, defects in cyclin D and cyclin D regulation will lead to cancer, right? Because you're going to have overproliferation occurring. All right. So RB, retinoblastoma, is the substrate protein of CDK4. So same thing, if there's a problem in RB or retinoblastoma, it's also going to then lead to dysregulation of this and maybe progression through the cell cycle when cells shouldn't. Okay. So RB first identified in retinoblastoma, hence why it's called that, which is a rare inherited childhood eye tumor. Okay. And so retinoblastoma or RB is a tumor suppressor gene. So if you inactivate that gene, so if that gene is mutated so that it is not functioning properly, it will lead to tumor development. It will lead to overproliferation of cells. It will lead to cancer cells. Okay. So it says proteins encoded by tumor suppressor genes like RB, right? And this CDK inhibitor, right, act as brakes that will slow down cell cycle. So normal functioning of these tumor suppressor genes slow down the cell cycle. Their normal function is to slow it down. If they are mutated so that they are inactive, you won't have that slowing down, and therefore then cell cycle will be increased. Okay? So it says RB plays a key role in coupling cell cycle machinery to the expression of genes required for cell cycle progression. All right. All right, so that was this is giving you an example of a tumor suppressor gene, and that this one tumor suppressor gene, RB, is actually so it says it's a substrate protein. So it's something that's going to interact, right? Its protein product of this RB gene is going to interact with CDK4, okay? And again, if it's mutated, it will basically cause increase in cell cycle progression. All right, so that was a tumor suppressor gene. Here, we're talking a little bit more about regulators of cell cycle, so DNA damage checkpoints. So remember, talking about those DNA damage checkpoints. And it says cell cycle arrest at DNA damage checkpoints is mediated by protein kinases ATM and ATR. So these are particular proteins that will be, or that are involved in arresting a cell when there is some sort of DNA damage that's detected. Now, again, you can imagine if there is a mutation or a problem in these proteins that are involved in regulating that, then cells will progress through that checkpoint when they shouldn't have. Okay, so it says these ATM and ATR will activate a signaling pathway that leads to cell cycle arrest. Okay, DNA repair and sometimes programmed cell death. I said this, right? If we think about those checkpoints, what the function is, they detect damage. If there's damage, halt everything so that it can be repaired. If it cannot be repaired, then the cell would go through apoptosis. Okay, what proteins are involved in that process? ATM and ATR. These are some examples. Okay, it says ATM will recognize double-strand breaks. ATR recognizes single-stranded or unreplicated DNA. So they're there to really recognize the actual problem. They are kinases, right? They phosphorylate and activate the checkpoint kinases, these guys here, that will then progress the cell through. Okay, so this is basically showing you this ATM and this ATR in terms of if they recognize some issue in the DNA, okay, they'll then cause that cell cycle arrest. Okay. Because they're basically going to be inhibiting this CDC25. So these proteins, the ATR and the ATM, are sort of the first players in terms of they're the ones that are actually identifying that DNA damage. They then will interact with other proteins downstream to basically halt that cell cycle. Okay, so we're not going to be able to activate that CDK to then progress those cells through. Okay, you can see that here. Okay, It says CHK1 and CHK2 phosphorylate and inhibit CDC25 phosphatases, which are required to activate, right? So that's what this is showing you here. These guys, once they become activated by ATR or ATM, they'll inhibit this process, okay? And so again, if this can't occur, CDKs can't be active, and basically cell cycle arrest, okay? So that says it here. Inhibition of CDK2 results in cell cycle arrest in G1 to S, okay? Inhibition of CDK1 results in arrest in G2. All right. So again, think of ATM and ATR as sensors, right? They're sensors for that DNA damage. So we already mentioned those checkpoints. We said, well, at those checkpoints, DNA damage, it's checked to see if there's DNA damage. How does that actually occur? Well, it's by two of the players are these specific proteins, ATM and ATR, that are the sensors to detect that. If they do detect it, how do they then halt that cell cycle? Well, it's by interacting through some of these proteins here. Okay, again, do we need to memorize this? No, but if I show you this, you better be able to explain some of the things I just talked about, okay? We should, though, know outright what the function of ATR and ATM is, okay? Okay. Now, in mammalian cells, okay, so now we're going to talk about mammalian cells. So, okay, here, you know, some more sort of mammalian cells. I will, well, so there are similar mechanisms in mammalian cells. This ATM and ATR were particularly well studied in yeast cells. Okay, let's move on though to talk a little bit more about regulation. Okay, specifically, it says arrest is also mediated by protein P3. Okay, so ATM and ATR are involved here. But there's also another protein involved in this process, okay, in mammalian cells, all right? So again, I'll tell you that a lot of what we know about how ATM and ATR work and what they signal through and what proteins they interact with, a lot of that work was done in yeast cells, but similar mechanisms exist in mammalian cells as well. One of the additional players or additional proteins involved in mammalian cells is called protein P53, okay? So it says this is phosphorylated by both ATM and CHK2, okay? So this is, again, in mammalian cells, here's another protein that's involved in this process that we just talked about. P53 is a transcription factor, okay? It says increased levels lead to induction of CDK inhibitor P21. So when P53's levels are increased, it basically leads to increase in P21, which is an actual inhibitor. If you went back to that table where I showed you those CYP-KIP families of inhibitors, you'll notice that P21 was one in there, okay? So P21 will inhibit this particular complex here, which will lead to cell cycle arrest, okay? And in fact, you know, you can consider P21 as being a tumor suppressor gene, right? It basically codes for an inhibitor, a CDK inhibitor. So any of these CDK inhibitors are, you know, the genes for those are tumor suppressor genes, okay? So this is showing you here now, you get a little more involved in specifically showing you the role of P53 and kind of how P21 is involved, right? So it says role of P53 in cell cycle arrest. So here's your ATM detecting that DNA damage. Basically, you're going to get increased levels of P53, which is a transcription factor, right? So it will bind to DNA and increase the transcription of P21, okay? P21 is a CDK inhibitor, okay? And so basically it will inhibit this complex. And so if this is inhibited, you won't have progression through. Okay, so again, you can consider P21 and really any of those CDKIs or CDK inhibitors as being tumor suppressor genes themselves. So anything that's going to slow down the cell cycle is a tumor suppressor gene. So it says here, P53 gene is frequently mutated in human cancers. Okay, and so if you lose function of P53, it will prevent cell cycle arrest, just like you see here, right? So if this is not functioning, this then can't act as its transcription factor to increase P21, it's going to lead to cancer, all right? So loss of P53 prevents cell cycle arrest in response to DNA damage. So that damaged DNA will then become replicated and be passed on. All right. So again, in a lot of different cancers, you'll see a mutation in this particular gene, this P53 gene. Again, P53 considered a tumor suppressor gene as well. Okay. So let me just kind of end. One other thing I just want to mention. I gave you a lot of examples of tumor suppressor genes. And so again, if you're going to mutate or if there's a mutation in a tumor suppressor gene, a gene that normally will slow down the cell cycle or keep cells from progressing through, like here, right? Like P53 and P21, these are tumor suppressor genes. If you mutate those and basically you have they don't function, right? Cells will be able to progress through that cell cycle when they shouldn't, right? When there's DNA damage. So let's just think just for 1 second, to think about, proto-oncogenes, right? If you think about genes that are going to code for things that will promote the cell cycle or promote transition from one phase to another, and basically there's a mutation in one of those that basically increases their activity. I guess maybe you can think of of just like a CDK in general. If there's a mutation in the gene that codes for a CDK, for instance, that will increase the activity of it and keep it active when it shouldn't, that could then lead to cancer as well. So for the idea of going from a proto-oncogene to an oncogene is that the mutation would cause the cell to, let me just do this because I feel like I don't know, maybe let me just add one slide real quick, just to make this point here. Okay. It would be awesome if it would write. What is this doing? Oh, it's not going to let me write, I guess. Okay, not in the video. All right, so anyway, a tumor suppressor gene is going to code for a protein that will normally slow down the cell cycle. And these are normal genes that exist in the human genome, right? That are normally there, just like this, what we just talked about. Let me go back, right? Just like P53 and P21, their normal function is basically to slow down the cell cycle or to stop progression if there is some DNA damage that's detected, okay? There are also normally in the genome genes that their job is to push cells through the cell cycle, okay? And we probably gave lots of examples of this as we're talking about kind of mechanisms involved here in moving from one phase to another. Okay, those are proto-oncogenes. So any gene that's going to code for a protein that will normally push cells through a phase or speed up the cell cycle are called proto-oncogenes. If you have a mutation in a proto-oncogene that makes that proto-oncogene more active, it then becomes an oncogene. And the reason why we would call it an oncogene is that it will then cause more proliferation than should occur. It will cause more progression through phases when that shouldn't happen, okay, and then lead to cancer. Okay, so a mutation in a tumor suppressor gene that basically suppresses its activity or makes it have no activity at all will lead to cancer. A mutation in a proto-oncogene that makes that gene more active will lead to cancer. Okay, so I hope that makes sense and you sort of can see the difference between the two. And I know, I've mentioned this before, but I feel like right now we got to just kind of put those two terms out there again. So particularly we talk more about showing the suppression, right, of it. And we didn't mention the proto-oncogene. So I just want to mention that term. So tumor suppressor gene, proto-oncogene, those are two types of genes that normally exist in the genome. They're both involved in regulating the cell cycle. Tumor suppressor genes normally will slow down the cell cycle or prevent cells from moving from one phase to another if there's DNA damage, right? Something like this. Proto-oncogenes will code for proteins that will speed up the cell cycle or are involved in progressing cells through the cell cycle. If you have a mutation in a proto-oncogene that makes that gene more active, it becomes an oncogene and can lead to cancer. Okay, so there's so much more we can say about this, guys, but I'm just going to leave it at that. There are some excerpts, summaries of different papers in the cell cycle chapter. There's definitely papers that we could read about that involve tumor suppressor genes and proto-oncogenes. Okay, we just don't really have the time right now, and so we're going to have to sort of leave it at that. But I hope we understand the relationship between the cell cycle and cancer. Okay, and actually, there's an entire chapter just on cancer, cell biology in the textbook as well. And so I would, if we had time, I would love to be able to go through that. But if you're interested, you could take a look at that, right? And so anyway, we'll leave it at that. Like I said, if we had one more week, I would love to cover that chapter, but unfortunately we don't. But I hope, again, we understand the relationship between the cell cycle and cancer, and understand that it's pretty complex. All right, thanks, guys.